Many proteins involved in antigen processing and presentation are encoded by genes within the major histocompatibility complex. The major histocompatibility complex is located on chromosome 6 in humans and chromosome 17 in the mouse, and extends over some 4 centimorgans of DNA, about 4 times 10 to the 6 base pairs. In humans, it contains more than 200 genes. As work continues to define the genes within and around the MHC, both its extent and the number of genes are likely to grow. In fact, recent studies suggest that the MHC may span at least 7 times 10 to the 6 base pairs. The genes encoding the alpha chains of MHC class 1 molecules and the alpha and beta chains of MHC class 2 molecules are linked within the complex. The genes for beta 2 microglobulin and the invariant chain are on different chromosomes. In humans, these genes are called human leukocyte antigen, or HLA genes, as they were first discovered through antigenic differences between white blood cells from different individuals. In the mouse, they are known as the H2 genes. There are 3 class 1 alpha chain genes in humans, called HLA-A, B, and C. There are also 3 pairs of MHC class 2 alpha and beta chain genes, called HLA-DR, DP, and DQ. However, in many cases, the HLA-DR cluster contains an extra beta chain gene whose product can pair with the DR alpha chain. This means that the 3 sets of genes can give rise to 4 types of MHC class 2 molecule. All the MHC class 1 and class 2 molecules can present peptides to T cells, but each protein binds a different range of peptides. Thus, the presence of several different genes of each MHC class means that any one individual is equipped to present a much broader range of peptides than if only one MHC molecule of each class were expressed at the cell surface. The 2 TAP genes lie in the MHC class 2 region, in close association with the LMP genes that encode components of the proteasome, whereas the gene for tapicin, which binds to both TAP and empty MHC class 1 molecules, lies at the edge of the MHC nearest the centromere. The genetic linkage of the MHC class 1 genes, whose products deliver cytosolic peptides to the cell surface, with the TAP, tapicin, and proteasome genes, which encode the molecules that generate peptides in the cytosol and transport them into the endoplasmic reticulum, suggests that the entire MHC has been selected during evolution for antigen processing and presentation. And that's many proteins involved in antigen processing and presentation are encoded by genes within the major histocompatibility complex. Have a great day.